Hello guys, welcome to our second part. Previously we looked at mitosis. Let's look at meiosis. And meiosis is a type of cell division that takes place in reproductive organs. We call them the gonads. And when this type of cell division takes place in the reproductive organs are able to produce gametes both in plants and animals. And this process results to gametes having half the number of chromosomes that in the mother cell or the parent cell, and we say they are haploid in number, meaning half the number of chromosomes as the parent or the mother cell. Then the principle underlying meiosis, and this meiosis consists of two successive cell divisions designated as Meiosis I and Meiosis II. In Meiosis I, the number of chromosomes is reduced by half or one half, and for this reason we call it reduction phase or reduction division. And the homologous chromosomes we talked about get separated from each other and go into different cells. And meiosis II, it results into separation of the sister chromatids. Remember, the first meiotic division, separation of homologous chromosomes. The second meiotic division, separation of the sister chromosomes, and for that reason it's called equatorial division. As you can see the summary here, the parent cell contains 4 chromosomes. As you can see here, these two are pairs. The homologous chromosomes pair together, and the first meiotic division, that is meiosis I, as you can see, the homologous chromosomes separate from each other. They separate from each other, as you can see, and move to different cells. Then second meiotic division, each chromosome separates to 2 chromatids, one of each kind going into each daughter cell, as you can see here. That's why you say the haploid state or haploid number. Then process of meiosis, each of the 2 meiotic divisions is divided for convenience into prophase, metaphase, anaphase, and telophase. And each stage is followed by 1 or 2 indicating whether it is meiosis I or meiosis II. Let's look at meiosis I. And this is early prophase 1, and the chromosomes shorten and thicken. As you can see, the chromosomes have become short and are very thick. When you stain, you can be able to see them clearly. Then on our diagram below here, showing the chromosomes, 2 chromosomes of the same kind called homologous chromosomes, and they form a pair we call bivalent pair. Let's move on. Let prophase 1, homologous chromosomes pair and lie side by side in a process called synapsis. And this is very important for you to know. They're laying side by side in a process called synapsis. As you can look here, you see here, they've laid side by side, and that process we call it a synapsis. And they form a pair we call a bivalent pair. This is a bivalent pair. This is a bivalent pair. You can look at the bivalent pairs, as you see. And each chromosome splits into 2 chromatids except at the centromere. And the contact between bivalent loosens and the chromosome moves away from each other, and however they still retain contact at certain points called chiasmata. And the chiasmata are basically the points of contact. In case it is one, we call it a chiasma. As you can see here, you have the point of contact of these chromatids, of the chromosomes. That is a chiasmata. And the chromatids of bivalent are seen to have crossed over each other, the chiasmata, and during this crossover occurs, and that is exchange of genetic material. Exchange of genetic material. Let me take you back. We have points of contact or point of contact here, that is chiasma. And in this point, the genes can be exchanged in what we call crossing over. It means the genes being different, because we said the chromosomes with the chromatids have different genetic constitution, and when they coil around each other, there is exchange of genetic material, leading to what we call genetic variation, which is very important. It is very important for the offspring. That is about Process 1 of meiotic division 1. Very important points about the chromosomes pair, which we call homologous chromosomes. They form a pair, we call it a bivalent pair, and they lie side by side in what we call synapses, and these chromosomes may coil around each other at points of contact, for them chiasma, and we may have exchange of materials, that's genetic materials, leading to what we call genetic variation. As you can see, the bivalent pair is here. Then chiasma, that is a singular one point of contact, is here, where there is exchange of genetic material. Then our next stage is metaphase 1. And the metaphase 1, the spindle fibers are fully formed in that, and attached at the centromere. As you can see here, they are fully formed. The bivalent moves to the equator of the spindle, and they do this as a unit. You can see they are still a unit, the bivalent pair. You can see this other side, they are still as a unit. And the sister chromatids orient towards the same pole, and the homologous chromosomes orient towards the opposite poles. And this positioning is different from that seen in mitosis. This will help you not to confuse between metaphase and metaphase 1. It will assist. I'll repeat. This bivalent pair, they move to the equator of the spindle fibers as a unit. As you can see, they are on one spindle fiber, the same to these other ones. And the homologous chromosomes orient towards opposite poles. Let me take you back to metaphase of mitosis. We said the chromosomes align at the equator of the spindle fibers, and they are independent. Each chromosome attached to its own spindle fiber. But in metaphase 1, they align at the equator of the spindle fibers, and they still occur as a bivalent pair, as you can see. The anaphase 1, homologous chromosomes separate, as you can see. They separate from each other and migrate towards the opposite poles. The sister chromatids no longer remain in parallel alignment, but diverge from one another. And the sister chromatids don't separate from each other at this stage. As you can see, these chromosomes are moving towards opposite poles because the bivalent pair are separated from each other. Then next stage we have telophase 1, and once the chromosomes reach the poles, they become densely packed together, and the spindle apparatus break down and disappear. The nuclear membrane reappears and surrounds each set of chromosomes. As you can see here, the nuclear membrane has reappeared. Then the cell then divides into two, like in mitosis. The daughter cells may undergo a short interphase, which we talked about before proceeding to meiosis II. And at the end of the meiosis I, separation of homologous chromosomes have been achieved. And when we say the daughter cells undergo a short interphase, we say the interphase, there is multiplication of genetic material, or replication of genetic material. They also said formation of new cell organelles, that is, for example, mitochondria, and maybe the Golgi apparatus. Let's move to meiosis II, and it aims at separating the chromatids from each other. Previously, we said meiosis I aimed at separation of homologous chromosomes, but meiosis II aims at separation of chromatids. First stage, we have prophase II. Chromosomes become shorter and thicker, and a new spindle fiber is formed. As you can see here, we have two daughter cells which were formed from meiosis I, and we're saying new spindle fibers are formed because the centrals have moved to the opposite poles. Then metaphase II, chromosomes migrate at the center of the cell, and they are at the equator of the spindle fibers. As you can see, the equator, that is at the center of the spindle fibers, and the spindle fibers attached to the chromatids, as you can see. Then anaphase II, sister chromatids separate and move towards the opposite poles. As you can see here, they are forming letter V because they are migrating towards the opposite poles. And the chromatids become the chromosomes of the daughter cells. Telophase II, spindle apparatus disappear. A new nuclear membrane is formed around each set of chromatids. As you can see here, the chromatids uncoil and regain their thread-like form. Then the cell divides into two. Thus, meiosis results into 4 daughter cells, each with haploid set of chromosomes. You can see 4 daughter cells. Then significance of meiosis, and it occurs in the formation of gametes like eggs or sperms, each with half the number of chromosomes present in the parent cell of an organism, thus carrying out sexual reproduction. And during fertilization, the haploid sperm fuses with the haploid egg to form diploid zygote. And through mitosis, zygote undergoes cell multiplication, eventually forming an adult organism with diploid cells. And a point to note, if the number of chromosomes were not halved during gamete formation, there will be a doubling of chromosomes for each successive sexually reproduced generation. Hence, this meiosis helps to restore a constant diploid chromosomal constitution in a species after fertilization. And one thing you need to know, meiosis provides opportunities for new combination of genes to occur in the gamete cells, leading to genetic variation in the genotype and phenotype of the offspring produced by the gamete fusion. And that is due to the exchange of genetic material during prophase I, where the chromosomes might coil, that the genes from each chromatid may exchange, thus leading to genetic variation. Let's look at the similarities between mitosis and meiosis. Both take place in plants and animals, and both involve multiplication of cells. Then differences: meiosis takes place in 2 phases, each having 4 stages. That is prophase, metaphase, anaphase, and telophase. While mitosis takes place in 1 phase of 4 stages: prophase, metaphase, anaphase, and telophase. And the 2 phases in meiosis, we said meiosis I, meiosis II. Meiosis occurs in sex cells or reproductive cells, leading to gamete formation, while mitosis occurs in somatic cells. We said somatic cells are basically body cells, leading to growth. Meiosis, homologous chromosomes associate, that is during prophase 1. Mitosis, homologous chromosomes do not associate or come together. Meiosis, we have chiasma formation and may lead to crossing over, hence variation, while mitosis, we don't have crossing over because there is no chiasma formation. Meiosis produces 4 daughter cells which are haploid, while mitosis produces 2 daughter cells which are diploid. Meiosis involves sexual reproduction and mitosis involves asexual reproduction, for example, binary fission in amoeba. That is the end of meiosis. I hope you've understood. You can like, share, comment for further questions. Thank you.